In 1961, Marshall Nirenberg and J. Heinrich Matthaei wanted to decipher the biological mystery and crack the genetic code. In order to do this, they needed a cell-free system to build amino acids into proteins. To do this, they put E. coli cells in a blender to create an extract of cytoplasm and ribosomes. For each trial of the experiment, they added amino acids to a test tube. 19 were labeled non-radioactive and 1 was labeled radioactive. This helped to track which amino acids were going to be made by which strand of RNA being put in. Matthaei then added a synthetic poly-U mRNA strand, incubated it in 37 degrees Celsius, where translation took place. After that, a protein was precipitated. The protein was then filtered, and the filter was checked for radioactivity. If the homopolymer coded for the radioactive amino acid, the resulting polypeptide would be radioactive. After many trials, it was found that the homopolymer coded for a chain of phenylalanine. The experiment was then recreated 20 times, with each tube containing a different labeled amino acid. The technology at the time only allowed the scientists to create homopolymers, such as UUU or CCC. They found that UUU makes phenylalanine, CCC makes proline, and AAA makes lysine. But they also found that GGG does not work. Another experiment in 1964 occurred by Marshall Nirenberg and Philip Letter. There was new technology, a filtration machine, that allowed for all codons to be artificially made, not just homopolymers. The first step was to make artificial mRNA codons and mix with ribosomes and amino-acylated tRNAs, one of the tRNAs still being radioactive. So you can see here, this is the tRNA that will bind to the ribosome to match with the mRNA strand, while these are the two discarded tRNAs. The next step was to filter the solution through a nitrocellulose filter. The tRNAs paired with ribosome-bound mRNA stuck to the filter, and unbound tRNAs passed through it. The filter was then assayed for radioactivity to determine the amino acid that was bound to it. The reason these scientists were able to determine the amino acids for a large number of codons was specifically due to the binding of tRNAs to the ribose-bound mRNA. This experiment helped to identify 61 different codons and their associated amino acids.